Okay, so just a quick reminder for anyone that brought any demo stuff or still has demo stuff sitting in their bag. I think Courtney from Parkindale said she might have some stuff. Not sure? Okay. Depends what fit in the luggage, basically. Yeah, so it'll be Geoff, and then we were gonna, if anybody wanted to discuss, maybe we'll have 5 or 10 minutes to talk more about controlled mass pollination tools and just suppliers, things that work, things that you've realized don't work, and then we'll do a little bit more on the protecting guards and seedlings. So Geoff and Stefan have some more items, and anybody in general just has had ideas during the day, it's a great time to share it. I've got a few slides just as I think the picture's a little bit better on some digital moisture monitoring tools, so people that can't afford water activity. I've tried and tested just about anything that you can use, so, and some little mini seed banks. We've got some microscopes and dissection tools. If somebody wants to see a pawpaw or cut a coulter pine seed or look at a red spruce, I dare you, you can. And then Victoria Lee's gonna give a little bit of a demo on germination testing, so little like 5 to 10 minute little brainstorms, and welcome Geoff from Kalmalka Research Station.
Oh. Sounds like, should I wait for Dave to finish rounding people up out there? I'll just get started. Okay, thanks everyone for sticking around to listen to me wrap up the day today. My name's Geoff Bradley. I am the seed orchard pest and plant health biologist with the Ministry of Forests. And that's a really awfully long title, which usually ends up getting stubbed down to I'm the bug guy. But more and more lately, and in particular for today, I'm the tool man. Today I'm going to talk about seed orchard monitoring. And I kind of got stuck into the low-tech tools, but in fact I'm going to drift probably more into the high-tech realm today with a few little low-tech things that we toss in there to include in our monitoring. And so yeah, I'm going to be looking at a salute to science and thinking about data to decisions. So when we're thinking about tools in the orchards, really the mantra that we've taken on in the last few years with the Ministry of Forests orchards here is this data to decisions idea. And this comes from a couple of veins that we look at this. One is just using the best information that we can to make the decisions that we're going to do for all of our orchard management. But the other big thing with this data to decisions way of looking at stuff is it goes away from the, taking the handful of soil and using your 35 years of gut experience and knowing that I need to water for an hour today, to being able to put tools in the hands of people who are new in the industry to make good decisions to manage their orchards. And the main things I'm going to talk about today as far as monitoring are what we're doing for water monitoring, nutrition monitoring, and pest monitoring. Realize there are other realms beyond that, whether it's cone counts and phenology and those sorts of things, but a lot of those are very regionally specific or regionally mandated, and there's only a limit to how many things I can talk about today. So when we're thinking about these kind of 3 realms of nutrition monitoring, moisture monitoring, and pest monitoring, there's a number of different types of data that we're collecting. Whether it's pest monitoring information, foliar samples, fertilizer records and fertilizer information, soil sampling, telemetry for water and moisture monitoring. But we look at them all and pull different aspects of them to kind of address different parts of these questions. So the first thing I'm going to talk about is our program as far as nutrition monitoring. So the big picture things that we look at and that we've been working on to improve our nutrition in our orchards. So in the BC seed orchards, BC Ministry seed orchards, that is to say, we've been using essentially 2 major tools for nutrition monitoring. The first is a fertilizer template and planning tool that we use to kind of figure out what we're going to do every year, and then a quick tool to record all of that data. And then we have an assessment tool that we use to visually compare our fertilizing records, our fertilizing history to our foliar results, and pull it all together. That tool uses, mostly based out of Excel, and it uses the Power Query functions that are built into Excel, which are really, really powerful tools for pulling a lot of data together into manageable data sets and preparing it for visual display. And more we're also starting to include soil sampling results in this, this toolkit. So the first thing that we use is this fertilizer planning tool. It takes into account all of our statistics for our orchards, whether it's the size, the number of ramets, and the spacing. We are able to, at the top, we set our targets for what we're aiming for our macronutrition for the year. And then we're able to break it down into our fertilizations and how they're going to contribute to that overall target for the year. And we can adjust that and kind of come up with a plan of attack that we're going to use for our fertilizing. From there, we have a really simple tool that goes into to just record what it is, what we put on what orchards. And this has been a really big step forward for us over the years. We've kind of had 5 different sites all operating completely independently, recording their data independently in different ways so that we couldn't really compare very nicely between sites, and we couldn't really easily see what other folks were working on. Now this is all put into the same format and kind of standardized so they can easily be used with these Excel tools moving forward to upload them automatically into our next tool, which is kind of the biggie. This tool takes all of the compiled data results from our foliar samples that we collect every fall, which are submitted in a standardized format from our lab, takes our fertilizer results and pulls them together to be able to kind of graphically look at them and see what are our foliar results looking like on the top here, and you can control the time scale that you're looking at. And what are our fertilizer applications looking like? And how are we seeing any responses? Are we seeing changes? And taking into account our kind of nutritional thresholds that we're aiming for to help us really guide with a targeted approach what it is that we're going to be aiming for our nutritional results next following year. So more and more, we're also starting to include soil sampling results into here. And in particular, the last 2 years, we've made a really big push to look at soil sampling in all of our orchards. This has kind of been a part of the missing link to really understanding what's going on nutritionally. And there's a lot of good reasons to do it. Understanding the physical makeup of our soil, understanding what's going on nutritionally, all the chemistry behind it. But then the big one in BC is regulatorily. There's a requirement from the Ministry of Agriculture, or Environment rather, that by 2025, most of our sites are going to need to do soil sampling as well. So the big thing that we're using as far as one of the tools that we're using as far as planning our soil sampling is information on soil sample or soil types. We do 1 composite sample per polygon. Our polygons are set using a combination of orchard layout as well as site characteristics and considering orchard management, the soil type, the topography, and using those to kind of set up the best subsample or soil sampling unit to be used to collect these samples. 15 to 20 subsamples, and we follow actually a lot of the BC tree fruit standards or practices for soil sampling because it's a very similar industry working in a very similar area and asking a lot of the same questions that we're asking. This is publicly available British Columbia Soil Information Finder tool, just an example of some of the diversity in soil types that can exist within orchards. You heard Rob talk this morning about the breakdown of that orchard that spans a couple of different soil types. This is an example from Vernon Seed Orchard Company where you can see sandy loam at the top, moving down to clay as you go down the bottom, and taking into that the knowledge from working on those sites to help develop what is going to be a good sampling polygon based off of your site characteristics is really important. Kind of the types of things that we're looking at, we've kind of broken it down into 3 sort of categories of when we're going to be doing our sampling. First would be baseline stuff that you would ideally be doing right before you plant any orchard when you can actually do the most about it. So water retention curves, soil texture, these are the things that you're going to need to know to start off. And then we're going to be looking at pH, cation exchange capacity, carbon. Those are things that we're going to look at every once in a while. They're not going to change really quickly within our orchards, but we're going to want to monitor over time how they're progressing. And then kind of annually or every couple of years taking samples and looking at our nutrient levels. And in particular, ammonium and nitrate are ones that, and phosphate are ones that are legislated that we need to look at, going back to those regulatory requirements. But then looking at changes in our micronutrients as well over time. So as far as nutrient monitoring, really what we're taking an approach now is trying to pull together all of these different ways of looking at nutrition in the orchard and looking at a big picture look at them. So we've been collecting foliar samples going back for years. We've got our fertilizer records for many years going back, looking at what we've been putting on the soil. Now we're starting to layer in thoughts about our soil results and how is that going to impact on our fertilizer plan, as well as crop size. The picture here is taken from, or the graph here is an example of some of the soil work that we're looking at in the last year. This is taken from our Kalmalka seed orchard, and this is just soil phosphorus concentration. And you can kind of see green, yellow, red, really easy to understand. Green, this is kind of nice place to be. White is you're too low. Yellow is where we're getting into the, we've probably got more than we need. And red is like, holy cow, this is kind of crazy. And where this really comes in is, as an example, looking at some of these soil results, you can see we're quite high in phosphorus in a lot of our orchards at Cal. And as a result, we were able to cut that aspect of our fertilization out for this year, which saved us several $1,000s on fertilizer costs right then and there. But it also helps us to kind of keep the soils healthy and follow the soil nutrition requirements that we are regulatorily required to do. The final thing looking at is crop size. I don't have a graph about this one here, but in the last year we've also started some preliminary looks at what is the nutritional impact of a crop actually look like. And I've done some of the calculations on what the impact, kilograms per tree coming off as cones and seed and going off to the tree seed center, how much are we actually pulling out? And that can, I think, anecdotally and by gut, we all know that a big crop is going to have a lot of nutritional impact, but we can actually get a bit of a read on that, and it's starting to be able to put some numbers to how much does a certain crop level take in nutrition, how much more do we need to put into the trees. And that's kind of an area that we can start to layer on over time. So going from nutritional monitoring, the next thing I'm going to talk about is soil moisture monitoring. So in this case, we're combining information that we're getting from our soil samples, as well as information that we're getting from ongoing soil moisture monitoring that we do in the orchards. The soil moisture monitoring we do using data loggers. We have one set up per orchard in each of our facilities. So some of our facilities may have 10 or 12 of these out. Some just have 1 or 2. Most of this work was spearheaded by Kate Hengel, getting this kind of up and running and into a really usable format for us. What a typical moisture monitoring station looks like for us is a data logger connected to 3 or sometimes more data probes. Rob talked about this a little bit in his talk this morning. Typically, those probes are set up with volumetric water content sensors, for example, in blue there, at 10 and 30 centimeters, as well as usually a soil matrix potential sensor set up at about a 30 centimeter level, kind of paired with the volumetric water content. And then at each of our orchard facilities, one orchard has an Atmos 14 atmospheric monitor that logs temperature, relative humidity, and barometric pressure. So just really briefly, I'm going to talk quickly about the two ways to measure soil moisture. First is volumetric water content, so how much water is there per unit soil? That's one thing we're looking at with the volumetric water content sensors. So just how much is there. But then the second question, and Rob also had the same picture that we both stole from the internet this morning, is how hard is it for the tree to pull that water out of the soil? That's what we're looking at with matrix potential. Across soil types, permanent wilting capacity and field capacity will change with their volumetric water content, but they're typically pretty standard at around negative 33 kilopascals and negative 1,500 kilopascals when you're looking at matrix potential. And that's really important because what we can do then is fine-tune, use that information paired with the volumetric water content to fine-tune our moisture regimes. This is an example of a water release curve. So on the x-axis, we've got the absolute value of pressure, so how much does it take to suck the water out of the soil. And on the y-axis, we've got volumetric water content. And every soil will have a curve something like this. Rob pointed out this morning, we've got a field capacity and a permanent wilting point. We can use this graph to set for each of our orchards what that permanent wilting capacity field capacity and permanent wilting point is. and we know our available water content. Rob also mentioned this morning, we want to stick, whenever possible, we want to try to target that 50% available water holding capacity. Here's an example taken from soil samples from lab measurements of 2 different orchards in our system. This top one, I believe, is from Saanich, so a heavy clay soil where the volumetric water content for field capacity is nearly 40%, and permanent wilting point is about 22%. The bottom one is from Prince George, where the field capacity is 18%, and the permanent wilting point is 12%. So very different, and knowing not just the number for the volumetric water content, but also the matrix potential can be really very useful in accurately figuring out your watering regimes for your orchards. You can also take the same measure. So I mentioned that we have the 2 types of sensors at 30 centimeters at most of our sites, and so we can plot, if you plot the matrix potential from the sensor and the volumetric water content from its paired sensor, you get a very similar graph. These are both, the top one is from Bailey Road Seed Orchard here in Vernon, and this one's from Saanich, same story. The water content alone doesn't tell you enough of the story, and the volumetric water content alone isn't giving you the complete picture, but when you look at the 2 of them together, you can really get a good idea of it. So we use Power Query again to pull all of our data logger information to look at our moisture content. So here's an example of one of our screens for Kalamalka Douglas Fir Orchard, looking at our matrix potential. So the bottom number here, it's hard to see in the graph here, but that's about minus 600. So we're well above that 1,500 that would be the permanent wilting capacity, keeping it nicely in the top half of our available water content. Here's a similar graph, but this is the graph looking at volumetric water content. You can see each of these peaks is one of our watering events. And using the information that I've just talked about, we can plot our field capacity and our permanent wilting point and set our targets for our irrigation. And we can use this to adjust the timing and the duration of our irrigation to make sure that we're keeping it where it needs to be and that we're not overwatering as well as not underwatering. Now what's really cool is here's the cloud version of it. So in one of our sites, this is from Cobble Hill, which is a remote site on Vancouver Island. It's run by our Saanich orchard. It's about an hour away. We have one of these set up, and this is cellular data that we're getting all of this. You can log in from any computer anywhere, see what's going on, and you can see volumetric water content on top, matrix potential on the bottom. So kind of the high-tech version of reaching in there and grabbing that soil and seeing whether you need to water it or not. Okay, so moving from moisture content, we'll talk about pest monitoring. So we do a bunch of different types of pest monitoring in the orchards, whether it's visual surveys, cone dissections, pheromone trapping, predictive models for things like spruce cone rust. And one of the big advances that we've made in the last few years is how we go about compiling and collecting all that data. But before we get there, I'm going to divert into my low-tech tip, which is one of my favorite little things that we've started using in the last few years in the orchards. I've got a demo of it over there. Pheromone trapping, particularly for Dirycteria, all of the work says you want to have the trap as high as you can, top third of the tree if possible. Well, without climbing a ladder or getting a lift out, it's really hard to get it up there unless you use one of these nifty little hangers that we fabricate up for a couple cents a piece, and a nice little pole, and it makes it slick and easy to get your, come on, Geoff, to get your trap in and out of the tree. You can pretty easily get it up to 15 feet up there, and they hang in there really well, with the exception of if you put them someplace where a real gun will bash them. That's the one thing you kind of have to be careful of. Okay, so that's the low-tech side of pest monitoring. On the high-tech side, a lot of what we've switched over to is using ArcGIS Online for our data collection. Survey 123 primarily to pull all of our data using custom-built surveys, but also a tool called Quick Capture and field maps to view our data, and dashboards that we can then pull it into to summarize. So Survey 123, you can use it on any tablet or phone. You pick your survey, you enter your data, and it's really nice because it standardizes the data collection. There's no transcribing it, anything like that. It goes straight into the database, and it's in a controlled way. Here's an example of some of the survey data from an intelligence survey that we're able to generate on the maps. This is Bailey Road Seed Orchard, and we can pull this data up. We can export it into Excel and work with it in all sorts of neat ways. We can set up dashboards so our orchard managers at a glance can see how many surveys have been done, what we've found, get a quick look, and then drill down into some of the details, for example, with our pheromone trapping. This is really handy to just be able to, so when the people who are doing the surveys are not the people who are looking at the data, they can kind of take a quick glance at this. Another tool that we've been using to put that data into ArcGIS is Quick Capture. This one's really great for fast surveys. So this is, again, from Bailey Road. This is Ponderosa Pine Orchard. And using this, you can literally, as you're walking along at walking pace, you click which button you want. It sets and records that data into the database. And you're able to do a 1,000 Ramette orchard survey in a matter of a couple of hours really quickly and easily. And then you can do neat visualization things with it when you're done. I'm getting a little short on time, so I'll just briefly talk about phenology data. So we do collect GDD and look at that to help us understand what's going on, both from cone development, but also pest development. I'll just skip right past that one. Here's an example of a couple of our GDD versus pheromone trap catches to help us kind of understand what's going on. You can see Directoria on the left versus pitch moth on the right, understanding the GDD and knowing that we get a peak of flight right at around 500 growing degree days really can help us understand managing that versus a long duration flight from a Directoria. Another thing that we've been doing a lot of monitoring of in the last few years with my various co-op students, Emma, Abby, and Annie, is beneficial insect monitoring. So we use sweep nets, video of a sweep net, another low-tech tool, sweeping both the vegetative cover and the trees. Branch beating, so taking your anger out on the trees on a bad day. Both really useful tools for cheap and easy surveying of beneficial insects in the orchard. We've looked at pitfall traps, although those haven't been super informative, really. We catch a lot of ground beetles, ants, spiders, and earwigs. Not too much else in there. But what we found over 3 years of looking at it is quite a huge amount of diversity in our beneficial organisms. We, over the years, have seen changes in between site diversity and quite a bit of overlap between sites, but a fair bit of unique species between sites as well. I can get into some of this data in detail if anybody's interested. But we collected in the first 2 summers, over 3,900 observations of 17,000 insects. Annie's adding about another third of that, or another 50% on this year. And we found over 50 types of beneficial organisms in our orchards in various numbers. A lot of them are ants, earwigs, and ground beetles, but quite a diverse group of insects. And really pretty, if you've ever seen, Michelle Hall did a master's of pest management in the 90s that had put together a book of all of the beneficial insect species that are typically found in seed orchards, and we find all of them on a fairly regular basis. Another thing that we've looked at is starting to think about how can we increase our beneficial organism diversity. And what we found kind of as a rule of thumb overall, which is not too terribly surprising, is that our beneficial insect diversity tends to be the highest in the settings with diverse plant habitat. So where we've planted out fallow areas, we've seen increased number and diversity of beneficial insects. And where we have cover crops in the rows, one thing that you do see is this is some data taken from 2022 in VSOC in the areas where we had the green strips, the inter-row planting, highest levels of insect diversity and the highest number of different types of insect classes found throughout the year. And so with that, I will say thank you and open up for any questions you may have.
Questions? Anybody? Ian?
Thanks, Geoff. What standards or levels are you aiming for with your foliar nutrient analyses? What's your guiding kind of targets?
So most of our targets come from Rob Broccoli's work kind of from 2000 to 2012 for forest stand nutrients, with a little bit of information coming in from the Christmas tree industry. So we have a table where we have all of those.
So how does that forest stand information relate to cone production?
It's the best that we've got right now. So that's what we use. I think trees, if we're meeting them, they're healthy, and it seems to work. If we hit those standards, the trees seem healthy, and they seem to be putting together good crops. But certainly it's something that an area that could use more work, but that's what we've got, so that's what we use.
Okay, yeah, that's where we're at in Alberta too.
So for your survey 1, 2, 3, would you be willing to share that, the monitoring part?
Yeah, one of the limitations of Survey 123 is to really make the most of it, you have to have an ArcGIS account. I am more than happy to share my survey schemas to copy and to use, and really to put the surveys and make them publicly available is possible. If there's interest in it, it just has to go through the BC government approval process to get them out there and put them into public hands for surveys. But yeah, I'm more than happy to share what
we're still using pen and paper, and it's not going well. It takes forever.
Yeah, pen and paper takes, so it depends on the survey. Some surveys are super quick with Survey 123. Some surveys, Survey 123, actually in the short term, it's a little bit more work than just writing it down on pen and paper, but you never have to transcribe it. You don't get transcription errors. You get to set the format of what all the data is going to go into your database as, so people can't put comments where they shouldn't be putting comments. They can't put formulas in that are going to mess up your Excel sheet. So in the long run, it gives you better quality, easier data.
Any other thoughts? Anybody got good data ideas? Data management is a never-ending thing, it feels like.
Thank you very much.
Okay, thank you. Okay, so Stephen had a few more slides just on demoing some things. I had a few more slides as well, just before we kind of get up and mull around, and if people want to come in front of the booths, we'll still do a few little hands-on things, or, yep.
What's that? I'm not going to do that. So I just brought some slides. I've got 4 different cages there, and this will just kind of explain them a little bit. Just some nice pictures, caged cone. That's what happens when bears get into them. Picture of some real cones. Does this have a thing? It does. So this was the first iteration, hardware cloth boxed cages. Then they went to the plastic cages. These ones are called flat cages tied with hog rings. I don't really know what that is. Oh, these are the original ones. The best ones are, this is in, there's a thing called Nutcracker Notes, and it's just Google Nutcracker Notes. It's from the Whitebark Pine Foundation. Go to issue 35, page 19, and there's directions about how to use the most refined, best method. And I'd recommend you go there. That's it.
Yeah, and mine was kind of the same thing. I wasn't sure how the day would go, so I was just, oh, not that one, nope, the... Oh, that's, nope. Nope, I think it didn't transfer. Okay. I'll just, the real quick version of mine was just a good follow-up for some of the low-tech stuff, and what I was going to explain in my main presentation was the fact that we're working with a lot of difficult and desiccation-sensitive species. So exceptional species are those, it's a bigger framework now than just orthodox versus recalcitrant. Exceptional species are species that can range the gamut, and most of them sometimes it's because the handling procedures from the field to just getting it to the gene bank, sometimes that's one of the biggest factors. So we've been really encouraging people with these little data technology tools. So a couple of things I brought are a little make-it-yourself mini seed bank that's based on what the Millennium Seed Bank was using. So they would take a 45-gallon drum, ship it to Madagascar, get a bunch of seed collection groups to do that, and they were conserving their seeds temporarily for a month or so in these little mini seed banks with silica gel, and then had these little data loggers in them. So for small collections, for conservation collections, we're working with a lot of indigenous groups now, and they don't have sometimes a lot of funding, so I'm trying to find really low-tech stuff for them to use. And so we've built this little mini seed bank that can be shipped, or they can use it locally, and it's a mix of this sort of food security if they want to use it, or for wild species conservation. So it's like $200 in total with a little kestrel data logger that we've been using, and Robert said he was using kestrels, so that's great. They're super rugged. I found them through Lee Valley because they're for hiking and heat stress, and they'll feed you like real-time relative humidity and temperature data, and then you can log it and download it, and it's Bluetooth, so it'll work right through the pail. You don't even have to open it. And then we just brought a couple of little microscopes. I've got some little good phone camera things you can put on great for bugs and pests and zoom lenses. And then there was one thing that I was showing a few people I thought might be handy is there's new thermal cameras that are clip onto your phone. And just for watching maturation drying, you can watch literally seeds dry in front of your eyes. So if you want to see a little demo of that, I'll have that little, I've got it on my camera, or on the computer. So I was just going to share that. Yeah, so I don't know, if everybody wants to get up, we can, maybe let Victoria do her little demo, and then we can play with a few things if you want. If you'd like to see a baby pawpaw tree, I've got one cut underneath the microscope as well. So stand up, stretch your legs.